Dear students, today we are going to discuss the last part of our lecture, Digestive System and Physiology of Digestion in Fishes. It is my request to the students who are viewing this lecture, please have a copy and a pen along with you. The digestive glands present in fishes are pancreas, liver, and some other glands. When we talk about the pancreas, they appear as diffused gland in the bony fishes. In some teleost, the pancreas in adults is hepatopancreas, that is, the hepato means liver and pancreas, they are joined together, they do not separate in the later stage and hence they form the hepatopancreas. Pancreas has two digestive functions, one is exocrine function and other is endocrine function. The source of exocrine secretion into the intestine, it acts as the endocrine secretion of the hormone insulin and glucagon. Talking about the liver, it is bilobed or trilobed structure in bony fishes, united anteriorly. Gland is usually yellowish brown in color. The liver in fish produces bile, which is stored in gallbladder. It is a key storage of the food energy in the form of glycogen. Talking about the other glands, the mucosal and the submucosal lining of the fish contains some glands having digestive function. Such glands are absent in mouth brooders. As we all know that there are certain fishes which brood their eggs in the mouth. If such glands are present in them, they will digest the eggs, and hence such glands are absent in the mouth brooders. The glandular structure of the stomach comprises the gastric glands in the cardiac and the pyloric regions as well. The secretory cells of the intestinal mucosa comprise the goblet cells and the granular cells. Talking about the physiology of digestion, the digestion of fish concerns the breakdown of food by enzymatic process and in many cases by acidic reactions in the gut. The diversity of the food in the gut of the fish attests the variety of morphological and chemical adaptations that have evolved for digestion. The system is to dissolve the food by rendering them so that they can be absorbed and utilized for the sustenance of life. Some food particles are used for forming the body tissues. We call that dairy process as the anabolism, whereas some of the food is used for the producing energy, that is catabolism. So the food can be classified as energy food and non-energy food. The energy food consisting of the carbohydrates, proteins and lipids, whereas the non-energy food consisting of the water, vitamins, oxygen, etc. The digestion depends upon the physical state of the food as well as the kind and quantity of the enzymes secreted by the digestive tract. There is variation with regard to the enzymatic profile of the fishes. Fishes depend upon variety of the food according to their feeding habit. They are classified as herbivores, carnivores, and omnivores. There is a great diversity in the form of the digestive tract, as we have discussed in our earlier lecture, that the different kinds of the fishes, that is carnivores, omnivores, herbivores, have the different length of the alimentary canal, whereas it is shortest in the case of carnivores, whereas it is the longest in the case of herbivores, and it is in between, that is intermediate in the case of herbivores. So the so the digestive tract depends upon the type of the food eaten as well, and it is divided into different functional regions. The basis of the digestive function, the regions of the alimentary canal are divided into 6 different regions. The first is the region of the reception of the food, or we also call it as the region of mechanical breakdown of the food. The transition from food capture to the initiation of the digestive process in the buccopharyngeal cavity is well illustrated by winnowing, that is sieving, in certain surf perches. This very region includes the mouth and associated appendages, which includes barbels. These barbels act as the taste buds. The perches separate ingested food from the non-nutritive food materials. They select that very and throw off the other which is non-nutritive and thus eject the debris. The pharyngeal jaws of the fish also exhibit the functional diversity. in pattern of winnowing, such as Cyprinidae, Mugilidae, Cichlidae, and Labridae. The pharyngeal jaws are important in other ways for the initial phase of digestion. Some fishes also show diversity in the form of the teeth. Those which feed on the insect larva have the conical and the elongated teeth, whereas those feeding on the algae planktons have the molariform teeth and the rounded teeth. Some fishes show type 3 digestive mechanism, that is trituration in the pharyngeal jaws, and the most fishes which show trituration lack stomach, so that finally ground plant material directly reaches the neutral, slightly alkaline region, which we call it as the intestine. The second, that is the gill rakers, the second region is gill rakers, as we have discussed in our earlier lecture, that gill rakers are also modified according to the mode of the feeding in the case of fishes. They are strong, teeth-like in the case of carnivorous fishes, whereas they are very delicate and filamentous in the case of planktivorous fishes, and they are in between, that is they are short and stumpy in the case of omnivorous fishes. Third is the region related to the conduction and storage of the food. It occurs in esophagus and is often muscular structure of the digestive tract. The glands are rare in this region. Sometimes enzymes reported from the esophagus may be present because of the regurgitation from the parts. The esophagus contains a number of the mucus cells and functions as a lubricant to transfer the food particles from the mouth towards the stomach. This very esophagus is short and narrow in the case of herby and omnivorous fishes, but it is in the case of carnivorous and predatory fishes. The esophagus is much larger, distensible to facilitate the swallowing of their prey. Sometimes it is filled with air to float, for example in the case of Tetrodon. Region of internal trituration. The early digestion, we call it as the region of internal trituration and early digestion. This region of This is the region which we call it as the true stomach, but it may be absent in some fishes. The lower gut of many fish species and mostly carnivores contains true stomach. Those fishes which have a stomach and thus releases the enzyme in the stomach, we call that very stomach as the true stomach. Whereas in some fishes, the part of the stomach is not able to release certain kinds of the enzymes and mostly the pepsin, and those very fishes we call them as the stomachache fishes. There in that very case only bulb-like structure is present, and that is the intestinal bulb. The fifth part is the region of the final digestion and absorption. We call that very part it as the intestine. This is the region of the anterior intestine or portion of the or posterior midgut, and may overlap with the preceding regions. In some species, which do not possess true stomach, their anterior intestine is swollen to form a sac-like structure, which we call it as the intestinal bulb, as I have already told you. There are certain fishes wherein the stomach is absent, and in holocephali and dipnines also the stomach is absent. The sixth part is the region of the fecal formation. The region where the fecal matter is formed is rectum and is not conspicuous as in the case of terrestrial vertebrates. In some species of teleost, rectum is not demarcated externally from the intestine, whereas in some it is wider than the posterior region. The Moving on to the physiology of the fish, we should be knowing that some of the important enzymes which are present in the fish. The fishes which do have the stomach, a true stomach, they secrete the enzyme pepsin, which acts upon the proteins to change them into the peptides. The pancreas also release trypsin in the intestine and change proteins into peptides, whereas the chemotrypsin released by the pancreas in the intestine, they act upon the proteins and peptides and transform them into the smaller peptides. The pancreas also secrete carboxypeptidases in the intestine and change proteins and peptides into smaller peptides and amino acids. The aminopeptidases secreted by the wall of the intestine, it acts upon the protein and peptides and transform them into smaller peptides and amino acids. The pancreas also secretes some tripeptidases and dipeptidases in the intestine, and which act upon the tripeptides and dipeptides and transform them and change them, and their product is amino acids. The pancreas also secrete lipase in the intestine and change triglyceride and act upon the triglycerols and change them into monoglycerides and fatty acids. Pancreas also secrete amylase in the intestine and change starch into disaccharides. Whereas in the case of some fishes, intestine secretes disaccharidases which act upon the disaccharides and change them into monosaccharides. Pancreas and the gut microflora, they act upon certain chitins by the help of the enzyme chitinases and change them into the N-glucosamine. Gut flora also secretes some enzymes which act upon the cellulose to change them into the saccharides. We talk about the digestion. It starts in the mouth and ends up at the anus. Many fish which chew pharyngeal with the help of the pharyngeal teeth or similar structures also produce mucus while chewing. The tests of the mucus in few species or enzyme activity have so far yielded negative results. Likewise, esophageal mucus cells, when examined histologically, showed no sign of containing any enzymatic granules, although there are some reports of the gastric like secretory cells in the posterior esophagus of some. As we all know, in all those very fishes which have a true stomach, they secrete an enzyme called as pepsin, and along with the pepsin, HCl is also released. And we all know the function of the HCl, that it makes the environment acidic, it kills the bacteria, it, uh, it helps in the degradation of the proteins as well. Whereas the pepsin is the predominant gastric enzyme of all the vertebrates, including fish. Its optimal pH for maximal proteolytic activity has been reported in the case of many fishes, such as it acts very nicely at pH 2 in the case of pike and plaice, and at pH 3 to 4 in the case of Ictelurus, whereas it acts very nicely, optimally at pH 1.3 and pH 2.5 to 3.5 in the case of salmon and tuna. It is the early phase of digestion occurring in the stomach where the gastric enzymes are secreted into its lumen by the uni or multicellular glands of the stomach lining. The gastric mucosa of the stomach of the carnivorous fish produces protease enzyme, for example pepsin, which acts at the optimal activity at pH 2.4 for the proper functioning of the enzyme, as we have discussed in the earlier slide. As the enzymes need a particular kind of pH for the proper functioning. In the earlier phase, the digestion pH should be acidic, and then it should be neutral, and then it should be slightly alkaline. So the enzymes are distinguished by the pH optima. I am again repeating that for the proper action of the enzymes in the gastric region, the in the earlier phase of the digestion, pH should be acidic, and then it should be neutral, and then it should be alkaline, as distinguished by the pH optima. And when we talk about the acidity of the elasmobranch stomach is quite considerable, where total, that is free and bound acid, is 0.6% and 1% of the HCl respectively. It has been shown by a number of the workers that this acidity increases with feeding. Earlier, it was believed that the stomach acidity is due to the presence of the organic acid, but later on proved that it is because of the HCl. The production of the acid in the stomach has also been noticed in the case of teleost, but in comparison to the elasmobranch, the acidity in case of the teleost is less marked. A number of the workers have reported pH values in the stomach. For example, in the case of the flatfish, which we call it as the Pleuronectus, the acidity ranges from 2.4 to 7.6, which means which is the mean of 5.65 which is the mean of 5.65. In the case of Esox species, the stomach acidity ranges from 4.5 to 4.7, whereas in the case of Nandus, pH has been found to be 6.5 in normal feeding and 6.4 in starvation. In Colisa fasciatus, the stomach pH is found to be 5.6. In Notopterus, the stomach is weakly acidic. It is below 6.5 to 6.8. um, it is between 6.5 to 6.8. The acidity has also been found to be higher at the surface, which declines beneath. For example, in the case of Esox, the pH at the surface of the stomach is 2.4 to 3.6, but beneath the surface, about 3 millimeter beneath the surface, the acidity was less, and around it was 4.7. The acid in the stomach is bactericidal. It kills the cells of the food, which may be alive when swallowed. It also helps in the decalcification of the food and helps in the absorption of the iron, besides facilitating the activity of the pepsin. Low gastric pH, that is 2.2 to 4.3 in some marine herbivore fishes, has been shown to be effective as the trituration in certain cell contents of the algal species. Even the lower pH values have been recorded in the case of cichlids such as Tilapia niloticus, which is a herbivore fish. The mechanism with low pH causes cell lysis. In poorly The mechanism by which this very low pH causes the cell lysis is poorly understood, but it is believed to weaken the hydrolytic bonds of the algal cell wall. Low stomach pH also facilitates the release of the free amino acids from the detritus, which creates the acidic environment, which activates the you can say uh, which activates the pepsinogen, which is at first the proteolytic enzyme of the food encountered in the digestive tract. Other gastric enzymes have been proposed but not firmly identified. Caecinolytic activity with an optimum pH 4.5 was claimed for the stomach of the Salmo irideus, but in most cases it is probably from the exogenous sources. If the fish are like higher vertebrates, then the stomach wall also produces the hormone gastrin, which stimulates the gastric secretion. A lipase may also. When we talk about the digestion in midgut and the pyloric ceca, there are two sources of the enzymes for the midgut, the pancreas and the secretory cells of the gut wall. With the pancreas, perhaps the secreting the greater variety and the quantities of the enzymes in the fish. Trypsin appears to be the predominant protease in the midgut. Most authors have just tested for the proteolytic activity over the pH range of 7 to 11 and reported their results as the tryptic activity. When we talk about the carbohydrates, they have perhaps excited the most interest of all the enzymes, particularly because salmonids do not handle the large carbohydrates molecules as well. And many workers wanted to determine the reason that why these various salmonids do not handle the large carbohydrate molecules. Talking about the role of bile, gallbladder, and the liver in the digestion. The bile salts acts as the detergents and serve to emulsify the lipids, thus making lipids more accessible to the enzymes because of the increased surface area, allowing some lipids to be absorbed and digested as microdroplets. In mammals, about 80% of the bile is recycled through the liver and gall. Secretion of the gallbladder in bile, which contains fat emulsifying bile fats, along with other pigments, biliverdin and bilirubin, that originate from the breakdown of the RBC and hemoglobin. The bile salts not only help to hydrolyze the fats, but also to adjust the digestive juices of the intestine to proper alkalinity for the action of the digestive enzymes. Liver, in addition to the digestive function, also serves as a storage organ for the fats and the sugar, glycogen, and important vitamins like vitamin A and D are also found in the liver. Tuna and cod liver are rich in these vitamins. Thank you very much. This was all about the physiology of the digestion.